So thank you very much for the introduction. What I'll try to do now between now and your coffee break is explain to you why it is so important for us to get images of cancer and not just the beautiful maps or rugs that we've seen Eitan Domani producing. So, we've seen the diagram of the hallmarks of cancer, and this diagram is extremely important in understanding the holistic view of the disease, which encompasses genetic changes in cancer cells and how they interact with the entire body in order to create this deadly disease. I like to put these hallmarks on this landscape view, where in the valley we have our regular, differentiated, well-behaving cells. And then as we go uphill, we see the cascade of increasing complexity of cancer. And when we climb on this mountain, we get more and more cells. In fact, we go through orders of magnitude, many orders of magnitude, from the single transformed cell in which the genetic mutation, that primary singular event occurred that made it, or perhaps a series of events that led to the genetic change which transitioned it from being a normal cell into a cancer cell, all the way up to the clinically relevant disease. And one of the important things that we need to keep in mind is that this initial event of transformation is prevalent. In fact, all of us carry with us transformed cells. We live with that, and most of us would die with that without knowing about it. So as we try to push early detection further down the slope of this mountain, it's important to make sure that we're not picking up those non-relevant transformation and actually turning each and every one in the room into a cancer patient. On the other hand, when things go bad and actually there is this very steep rise in number of cells, it's important to predict that this is what is happening, understand where the critical points are in order to intercept and intervene. So imaging plays a critical role, and it has actually transformed the way medical care is being done. It is often the primary event at which your doctor would be able to tell the patient where there is cancer, what type of cancer. It's diagnostic, and it's also being used for critical decisions along the pathway for cure. Now, if we look at a picture like this one from Vic Muniz, it's important to see the holistic view, the whole body view, the whole physiology, as we've so beautifully heard earlier this morning. But it's also important to look at the details with high magnification. And this is what we need to do when we look at cancer. We need to understand where the disease occurs in the whole body. We need to understand changes that occur as consequence of the disease, loss of body mass, loss of muscle tone, degradation of bones, and so on. And we need to look at the tumor cell in the minute details and try to understand what is the event that is happening that's driving this cancer. Now, when we look at tumors, they contain a wide variety of cells. They include, of course, the tumor cells, but they also include infiltrating immune cells, fibroblasts, these are the cells that line most of the organs in our body, blood vessels, and a very small population of cancer stem cells, which are the ones that drive the growth of the disease. And in order to study those, we use a variety of imaging, which is the cell sorter, in which we take the whole tumor, break it down into single cells, and then we can get the distribution of what percentage of the tumor is taken up by each one of those cells. But this would not be sufficient, and you actually need a picture like this one, in which the tumor cells are in green, and in red you can see the blood vessels around that, in order to see the interaction between them and how these small tumor nodules are engulfed by the blood vessels. And only then you can start to understand the rules of the game. And the analogy I like to draw to that is a sorting of a very familiar scenario, at least for those of us on the other side of the Atlantic, where a cell sorting device would give us this distribution of grass and cement and perhaps some human DNA and some food material. And this would never tell us what is actually going on, but you would immediately be able to understand that something very important is happening if you use this streaming imaging device. Okay. So imaging is very critical and very critical in cancer. So what can we say with imaging except the position of the tumor? Well, these days, imaging is being pushed in order to be able to visualize specific events on the molecular landscape and on these hallmarks of cancer pathway in a very precise manner. And I'll give you an example of cell proliferation, where in order to generate DNA to sustain the proliferation of tumor cells, you need the building blocks of DNA, and you can label them with a radioactive tag, for example, and then this can be used in a PET scan in order to detect the tumors, look at the proliferation of the tumor cells, and within the tumor to see whether a treatment, which is supposed to be killing or blocking proliferation of the tumor cell, is actually doing its job. There are other changes in these hallmarks of cancer chart, and one of them is a very important change in the preference for food, in the preference of nutrients of cancer cells relative to normal cells. They have a sugar tooth. They like sweet things, tumor cells. They have a very high affinity to glucose. So if you provide nutrients of various types, the cancer cells specifically would take up very large amounts of sugar. And this is being used in the clinic in the FDG PET, which some of you might have heard about, and you can see it on the left side. Now, what you can see here is a patient with multiple metastatic lesions. If we wanted to biopsy and generate a DNA type array of each one of those metastatic lesions, that person would undergo extremely invasive, unbearable type of diagnostic. But in order to do that with imaging, you only administer this radioactive glucose, and you immediately see where the tumors are. And if treatment is now blocking this pathway, you would be able to follow the change directly. Now, in order to do this on a screening system, the radioactivity of PET might become a problem, and particularly when it's combined with CT, with computerized tomography, that's not an exam that can be repeated often on the same patient. But a new alternative, which is being evaluated now in the lab of Lucio Friedman at the Weizmann Institute, uses MRI in order to detect the same metabolic changes along the same pathway using non-radioactive imaging. And this can be used in order to monitor metabolic changes, changes in the preference for nutrients in cancer cells non-invasively. Another very important hallmark of cancer, its ability to invade and metastasize. And this is perhaps one of the changes that occurs in cancer, which is most devastating because it prevents success of treatment with surgical intervention. And one of the things that we want to see is the boundaries of the tumor and to see where the tumor cells are. You can see it here in purple in this ovarian cancer tumor as it infiltrates into the matrix. And when it invades into the matrix, it changes the matrix environment. And this is work which is being pursued in my lab, trying to understand how the cell interacts with the building blocks of the tissue. We do this in collaboration with Irit Sagi, also at the Weizmann Institute, looking at different proteins that affect the matrix in which the cell progresses. When we talked about the sugar affinity of tumor cells in ovarian cancer, which is the tumor that my lab has been studying mostly, the tumors shown here in red actually express on their surface receptors that can bind long chains of sugar, very much like this one, the sugar treat which the children love in fairs and circus. They bind, they use it in order to adhere to the surface, in order to metastasize. These long sugar chains are anti-angiogenic. They actually prevent the growth of new blood vessels, but the tumors, after binding to it, degrade these long chains of sugar, making them inducible for growth of new blood vessels. So tilting the balance from inhibitory to stimulatory, it's one of the ways by which tumors teach the environment to become favorable for tumor growth. And it is this education of the environment, this corruption of the environment, which is something that we need to study more deeply in order to understand how to combat cancer. So what we've done here is create a beacon that would detect the binding of the tumor to these long sugar chains. And when we do that, we can accomplish something which we think could potentially be important. So this is a slide, a microscopic slide taken live in a mouse, and it looks normal. You actually do not see a trace of a tumor here. This is what the surgeon would see when they are opening a patient. Wouldn't it be wonderful if the tumors could blink? So in an experiment, we can do that. We can make the tumors glow bright red. There are many tumors here. That's the way ovarian cancer metastasizes. It implants with very small nodules everywhere. And the way to treat it is to go for a debulking surgery in which the surgeon does its best to excise all the visible tumors. But they're right now only using their eyes, and they do not have this red glow to assist them. So if we stain in green these receptors that bind the sugar, we can now decorate all these little tumors with the green glow. And you can see how they co-localize. So with the green glow of this sort, we can create something that might help a surgeon do his job better. So we talked a little bit about growth of new blood vessels, the field of angiogenesis, and I would like to acknowledge the very important contribution of Judah Folkman. He talked in one of the previous global gatherings when angiogenesis was a huge hope for therapy. And this is one of the theories that led to immense work on angiogenesis. The thought is that angiogenesis sits at the critical switch point between tumor dormancy and tumor growth. So tumor cells proliferate, they form 2 cells, 4 cells, 8 cells, until they reach a size of 1 millimeter. And then they stop. They cannot grow anymore because diffusion becomes a limitation and they do not have enough oxygen or glucose to sustain further growth. And the only way they can grow, or so was the angiogenic switch hypothesis, is by induction of growth of new blood vessels. So I would like to show you a cartoon which was generated by Rakesh Jain at MIT, which explains this in more detail. And before we go into the movie, I'd like to introduce the players. So the key, that's out, yeah, I'm getting support here. Okay. So endothelial cells are the red ones which line the blood vessels, and the blood which is flowing within the tubes actually comes in direct contact with the endothelial cells. On the outer side, there is this jellyfish-type cell, the pericyte, which hugs the blood vessel, and these pericytes provide survival signals to blood vessels. They actually stabilize the blood vessels, making sure that our vessels that were here yesterday would be here today and tomorrow, and would continue to perfuse blood to all our organs. They have an additional important role, which is to constrict or dilate. And when they constrict, they actually reduce blood flow. When they dilate, blood flows to a larger extent. And this is the way blood flow is regulated to our organs. So right now, when I'm talking, the area in my brain which is responsible for speech, the blood vessels are dilated. And in your case, the area of listening, or maybe the area of thinking about coffee for the break. Now, in order to make blood vessels grow, you need to overcome very important stabilitory regulations. So the blood vessels are not supposed to change. They're stationary, and they do their function in regulating flow, but sometimes you need to make more blood vessels. For example, if you never exercised, and now you go to the gym and you build muscle, or on the other hand, if you stopped exercising completely and you just eat hamburgers in front of the TV, and now your fat tissue is growing, in both cases, you need to make more blood vessels. And the way to do that is the tissue sends out a signal called VEGF, which binds to its receptors, and this activates a whole cascade of signaling, instructing the tissue to build a new blood vessel that would nourish the new muscle or fat. So let's run this movie. So we can see here a normally functioning capillary. There are pericytes around it. They control the flow. If you need more flow, they would activate it. But now there is a signal coming. So VEGF binds to the receptor, and it sparks this signal into the cell. Now, in response to this signal, the basement membrane, a patch of it, disintegrates, and now the cells, the endothelial cells, become plastic, and they send a capillary, which is going into the tissue and sending blood to the area where it's needed. And this is a very well-regulated system, allowing each and every cell of our body to get enough oxygen. Now, this is a tumor, and the tumor forms lots of VEGF. It floods the whole area with very high signaling, actually like pushing the accelerator all the way down. And the blood vessels, they know what they need to do. They get the signal, they signal, and now they would respond with growth of capillaries that reach the tumor. And the tumor cells now are no longer starved, and they would start to grow and proliferate. Now, in a normal tissue, this is done in a very well-orchestrated manner, and immediately the pericyte would tag along and would stop the growth of the blood vessel. But the blood vessels in tumors are different. They're leaky, and fluids and proteins from the bloodstream ooze out from these vessels. So can we detect these particular changes in the blood vessels of tumor and use that as part of our diagnostic tools? So there are many questions that arise from this movie, and these are the ones that are driving research in my lab. We're trying to understand the cascade of events. We're trying to understand the players, and in particularly, we're trying to understand this lack of parasite in the tumor vessels and whether there are other cells that participate in the process in the case of cancer in the absence of this parasite. So looking at changes in vascular permeability in tumor is actually the way most cancers are diagnosed today, particularly with respect to brain tumors, but also in breast. And this is the work of Professor Hadassah Degani, also from the Weizmann, who developed an algorithm which allows you to differentiate between benign and malignant tumors just based on the dynamics by which material oozes out from leaky blood vessels in malignant tumors. And you can see this type of oozing out from tumors in the tumor that is seen on the right hand, in which you can see the material radiating out from the tumor to all directions. When we looked at this type of dynamics, one of the things that struck us is that actually material is not sustained within the tumor region where you want it to stay. So for example, if you administer those very active biological drugs through the bloodstream, or you administer chemotherapy through the bloodstream, the material actually oozes out radially to the normal tissue in all directions, sparing the tumor itself. And trying to understand that, we developed tools in order to look at the growth of lymphatic vessels, which drain the periphery of tumors. You can see them here in green. The tumor is in blue and blood vessels in red. And these are routes by which tumor cells can send metastases to distant organs. So the dynamics of fluid in the tumor actually teaches us about the way by which cancer cells can reach very distant locations and infiltrate and colonize to form metastases. Now, it's very hard to look at lymphatics, and we've developed some of the first tools in order to visualize lymphatic drain using MRI, shown here in green. So this would be another way to look at the metastatic potential of a tumor even before it started to colonize distant organs. So there are no anti-lymph angiogenesis drugs yet on the market, but when there will be drugs, we will be ready with a biomarker to find the patients who can benefit from those treatments. If we look at the distribution of material administered to the bloodstream, we can see it here in green. This is an ovarian cancer tumor, and we can see it in a region which is surrounded by red cells. You can see them faintly. The blue are the cancer cells, and actually none of these material which we want to deliver to the tumor ever reaches the destination. Actually, there is a shield around the blood vessels which protects the tumor cells. That's not what we need in order to treat cancer. And it's a resistant mechanism which is physical. It's in the structure of the tumor, and you would never be able to see that if you just look at genomics. You need the imaging, the distribution, the architecture of the tumor in order to understand what is happening here. So these tumor myofibroblasts, they're very prevalent in the tumor. In ovarian cancer, they can be about 50% of the tumor mass. Half of the tumor are these stroma cells. They're normal cells. They're not cancer cells, but they help the tumor vasculature survive and protect the tumors. So can we use that in order to detect tumors? We can see how they align around the vasculature, how they hug the blood vessels and provide the survival cues. And we can actually see how they home to the tumor systemically. If we talk about the systemic, holistic view of cancer, this is one of them. It's enough to have a tumor nodule in one part of the body in order to start to attract cells, immune cells, fibroblasts, and other cells from the entire body, and they home to the tumor. And in the tumor, they're corrupted to help the tumors grow. So why is this important? It's important because they create a barrier for therapy, but they can perhaps be used for detection and perhaps also for delivery of therapy. And this is almost fresh from the oven. It's a study in which we highlight or create a blinking situation, a sparkling situation for these very small ovarian cancer tumors, where now the signal is not a contrast agent that we delivered, but rather labeled cells that home biologically through the native mechanism into the cancers. And in the cancer, they reach the right place around the blood vessels where they're exposed to all the treatments. Now, we can follow that also by MRI. That's a study done in my lab very recently, and we're now collaborating with a hospital in Israel in order to check this on fresh cancer material derived from patients. That's the patient-derived tumor xenografts, in order to see whether a patient's own fibroblast could be used as an aid in order to improve the surgical debulking procedure, and perhaps later on also to deliver anti-cancer treatments. So looking at this cascade of tumor progression, what we would like to do is tile it with multiple imaging tools that would allow us to give a panoramic view of cancer development and assist patient therapy by providing the information needed for clinical decisions at all the steps of treatment, during the screening phase, after detection, during therapy, detecting early recurrence of tumor, and following patients as they move from one type of treatment to another, trying to guide the type of treatment that is being provided. Now, of course, all of this is a very important collaboration of many people. In order to do this type of work, you need to combine chemistry, physics, computer science, and life science. There's almost no other place like Weizmann where you can bring all these forces together on one campus. There are many students and many collaborators, and of course, many people here in the audience that helped to make this work happen, and I hope will help it move forward. Thank you.
Thank you, Michal. I think we all appreciate that panoramic view, but also the artistic aspects of your presentation. Really very nice. We have time for a couple questions. Someone? Let me ask you something. We know now there's a drug, an oral drug for diabetes, metformin. And that seems, if you're on metformin, to reduce your chance of developing a cancer. And I would think that your discussion of sugar and the need for sugar for cancer cells is one of the explanations. Do you know about this work?
Now, so we've been looking at the basic pathways. We haven't been following in my lab treatment strategies, but it would make sense that whenever you would intervene with a metabolic pathway, you would affect cancer, because metabolism and changes in metabolic pathways is so central to cancer progression.
Very good.
So it makes a lot of sense.
Yeah. Any other questions? Yes. Louise. Microphone, please. Right back there.
Thank you. I understand that one of the ways to try to fight the cancer cells is to make them commit suicide, apoptosis. Is there any development in this type of treatment?
Mm-hmm.
Yes, so there are definitely treatments that try to induce cancer cell suicide. The problem is, of course, always to differentiate between normal cells and cancer cells. So it's the same suicide pathways which are used by normal cells as well. So you would need to somehow deliver the treatment correctly. And one of the tools that we've been developing is actually to monitor cancer cell suicide so that we would be able to see that suicide is happening at the right location and not elsewhere.
Okay, thank you very much.